Okay, class, we're back. So we said the muscle cell is long. It has multiple nuclei along the peripheral surface. We said that occurred by muscle cells fusing together. It has many mitochondria. It needs a lot of energy. We already talked about the cell. Glycosomes store they're little vesicles that store glycogen for glycogen storage. Glycogen is needed for muscle contraction. Okay, need energy. We kind of talked about ATP before. Okay, we talked about how ATP would be formed. We talked about a little bit about the Krebs cycle, you may recall. Myoglobin is the red in muscle. That's the red stuff. It's the brother of hemoglobin, and it holds on to oxygen. See, muscle needs a lot of oxygen to make the ATP. Okay? It also contains the myofibrils, which we'll look at, the sarcoplasmic reticulum and the T tubules. All of this we're going to look at in a moment in the anatomy area of this. Okay? So we go further. The myofibril is inside, let's go here, is inside the muscle cell. So the myofibril right here is inside these are all myofibrils inside a muscle cell. This is the contractile component. This is where the contractile myofilaments are, right there. Okay? So you can see it's 80% of the cell volume. It's the majority. In skeletal muscle, it exhibits striations. Why? Because I told you earlier that in skeletal muscle and cardiac, the myofilaments, which are in here, are arranged in a myofibril pattern. Okay? So that's what we're looking at. So we come here. Here's that muscle fiber, myofibril. So we go a little further here. The smallest contractile unit of a muscle fiber is a sarcomere. So now this is where we get into this picture here. Okay. First of all, here are the things, and this is in my PowerPoint. These are the things that's present. These are molecules present that we'll talk some about in this muscle. Okay, so we'll talk about that a little bit. All right, now we're coming to a sarcomere. Okay, this is what I have to explain. I want you to read on it too. Right here, this is a sliver. Now I want you to understand what this is. This is a three-dimensional drawing of a myofibril. What we're doing to see this is taking a thin sliver right down the middle of this, because you can't see this under a scope, but you can see this, a thin sliver. So this thin sliver is this, and what it's showing is the interdigitation of the myofilaments. See, all of these things here are myofilaments making up the myofibril. All right. Now, the players in the game, and you're going to have to really study this, is something called actin that I'm going to show you the structure of. Something called actin. Something called myosin right here. The two major ones are actin and myosin. Right here, what will happen is these actins come together and hook right here. So from this, see, this kind of looks like a Z. From this area to this area is called a sarcomere. The sarcomere is the functional unit of a muscle fiber. You say, what's a functional unit? It's the least amount of anatomy that can explain all the physiology. It's the least amount of anatomy they can explain. So in other words, the muscle is made up of a bunch of sarcomeres. If I can explain how one of them works, I can explain the whole thing. All right. So what you have here is this actin. Now let's look at this. Okay, this actin right here. See if I can blow it up a little bit. So we're gonna come here. So you gotta study it also yourself. Right here, I'm coming in with actin. This is actin right here. These blue are actin. Okay? I'm going to come here and show you what actin is made up of. So I'm coming to this picture. Okay? Right here, what we're looking at, we'll bring this down just a little bit. Okay, actin is made up, so we start with a polypeptide, just a straight polypeptide. Then we're going to take that polypeptide and fold it into a ball. So we're going to take a polypeptide and fold it into a ball. And when we fold it into a ball, that's about 275 amino acids, don't worry about it. But when we fold it into a ball, it forms a globule, and that's what we call G-actin. It forms a globule. Then what we do is polymerize. That means hook up. So we're hooking up these G-actins. And then we hook up more and more and more until we finally come with a chain of this. And we're going to call that F-actin. Then I'm going to take two F-actins and wrap them around like a wrapped around pearl necklace. And that right there is going to be what we call actin. This is the actin that you see. Now I want to go back to the G-actin because we're going to need it. On every G-actin, there's going to be a little dot on every G-actin. Now I want you have to really go back and forward on this. And this dot is called the binding site. Right here, this dot, which is on every G-actin, is the binding site for myosin. So on actin, there's a binding site to hook myosin. On actin, there's a binding site to hook myosin. And these dots on the G-actin are that. Okay? So that's actin. So I come back here again. Come back here and see this is actin. So that right there. That's actin. Okay, I'm not through with actin yet. I'm going to come back to actin again. And I'm going to say, in addition to actin, there's a couple of other things associated with the actin. Now you can't see that in this drawing here, but I'm blowing it up. Associated with the actin, which is this, is something called tropomyosin. This ribbon thing is a protein too. And that ribbon thing is called tropomyosin. So take up a pearl necklace and you interdigitate a red ribbon in it. A pearl necklace, a double pearl necklace. So you wrap two pearl necklaces, then you interdigitated a ribbon through it. That would be the tropomyosin. A couple of ribbons. Then another thing you have is ever so often you have this we're going to call a troponin complex. A troponin complex is made up of three things. Each troponin complex is made up of three things. Those three things are a troponin I, a troponin C, and a troponin T. Let me say it again. A troponin I, a troponin C, and a troponin T. A troponin I, a troponin C, and a troponin T. All right. The troponin I, so here we go. The troponin I hooks to the actin balls. The troponin I hooks to the actin balls. The troponin T hooks to the ribbon tropomyosin. So I is hooking to the actin balls. T is hooking to tropomyosin. So immediately you can see tropomyosin is not directly bonded to actin. There's a liaison that's gluing it together, and that's the troponin. Okay, now I'm left with troponin C. Troponin C, which is going to be important, binds to calcium ions when they are available in the cytoplasm of the muscle cell. So I binds to actin, T binds to tropomyosin, and C binds to calcium when it's available in the muscle cytoplasm. And we'll come to it. So we go back again. That's the structure of this. I named everything. Now, right here, this is where we come in And I'm going to hook these together at this thing here. And the structure and another protein comes in to do that. And that is going to be called alpha-actinin. Alpha-actinin. Let me take it over. Alpha-actinin is going to hook these two like this. Now, really, if it was three-dimensional, this is surrounded by several of these. So, in other words, there are actually 6. See, this is a sliver. So, this is only 2D. It's hard to show you the 3D. Okay. So, in other words, this myosin is actually surrounded by 6 of these. 6 of these. Okay. Around there. Be this, then 1 here. So just think about this being surrounded by 6 of these. Because see up here, you can see that the deep red is surrounded by a lot of thin stuff. Okay. Now, right here where that alpha-actinin is, you see how this looks Z-like. Okay. So this is called the Z-disc or Z-line. The Z-disc or Z-line. And this is where the actin is hooked together. Right there. Between 2 Z-disc or Z-line is 1 sarcomere, which is the functional unit. Okay. Now, I need to hold this myosin. We'll go into that in a moment. I need to hold the myosin suspended, because otherwise it would just fall. So the myosin is being held by this right here, a protein called titin. Titin is holding this myosin to this Z-disc. Titin is holding this myosin to this Z-disc so that this doesn't just fall down. Over here would be another sarcomere. Over here would be another sarcomere. Okay? Now, we come in then, and now I want to look at myosin. So I'm going to come here. Here's myosin. Right here, myosin is 2 long proteins, 2 long polypeptides, shall I say, that are wrapped around one another. 2. Each one of them has a head. So this is 1 myosin molecule, and it has 2 heads. Each of these has its own head, but these are wrapped around one another. When you wrap them around one another, it looks like a golf club almost, a 2-headed golf club. Right there. When you wrap them around one another. Now, when we look at the head, so you got to study this. When we look at each head, there are 3 things that's going to come into play on each head. 3 things. Something called the actin binding site. Now remember, see this is for actin. Remember what we had? On actin, we had a myosin binding site, that dot. But on myosin, we have an actin binding site. The other thing we would have, which you don't see here, would be ATP. There's an ATP binding site on this. So that's the other thing you'll have is the enzyme ATPase. So 3 things on every myosin head. An actin binding site, an ATP binding site, and the enzyme ATPase. And ATPase is the enzyme that breaks down ATP when we need it. Okay? Now if I come back here, notice how these heads here, so I come back here real quickly, and what we have here, we're going to hook up a bunch of these. And each of them you're seeing these heads sticking off. Each of them you're seeing these heads sticking off. So when I come back here, those are those heads. Now the heads here in one direction and the heads here, because what happens is we're going to bind the 2 ends together by something called M-line protein. Okay, we're going to close this one and we're going to come back to this again. Okay, takes a lot of work on this anatomy stuff. All right.